So let's study cytoskeleton. The word cyto here, it means cell, and skeleton. So basically the skeleton of the cell we are trying to study. So in this cytoskeleton we have three things. So remember the number 3. So we have microtubules, we have microfilaments, and then we have intermediate filaments. So I want you to understand something, that these microtubules are bigger in size, and these microfilaments are smaller in size, and these intermediate filaments are intermediate. It means they are medium. Cool. Let's study about microtubules first. First of all, let's understand why they are named this way. Let's try to remember this. Think it like plumbing tubules versus electric wires. That's how I remember it. For example, I remember plumbing. Plumbing uses big tubules, and electric wires inside your doma, inside your house, it requires small filaments in the bulb, right? So these filaments are small and tubules are big. Just remember this. And let's study this as well. Just imagine this is one neuron. This is the cell body of the neuron. This is the synapse of the neuron. And this is the axon. So usually what happens is that cell body sends the message towards the synapse, and we call this forward movement. And if the movement of the message is from synapse to the cell body, we call it reverse. Just keep this thing in your mind. Okay, let's classify cytoskeleton on the basis of their sizes. Cool. Microtubules, big in size. Of course, what's their structure? Their structure is 9.9 plus 2. What is 9? What is 2? 9 is the outer doublet. What is doublet? In pair. So this is how it looks like. How many pair is that? 1, 2, 3, 4, 5, 6, 7, 8, and 9. And inside 2. That's it. These 2 and these 9 pair. That's how the microtubules are in structure. Cool. What are these things? These things are just proteins. Cool. Let's study microtubules, big in size. Just remember the number 3. They have 3 proteins. 3 proteins that are microtubules. What are they? Kinesin, dynein, tubulin. Cool. What are their function? The first function is axonal transport. This one. The second function is ciliary motility. And the third function is movement of the chromosome. That's very important, Cool. So the first is kinesin, second is dynein, and the third is tubulin. Cool. Let's start with the first function. Axonal transport. Forward, going from cell body to synapse. It can be fast or it can be slow. If it's fast, it's 400 mm per day. Or if it's slow, it's 0.5 to 10 mm per day. It's slow. This is fast. Cool. What is reverse? Reverse is fast. How fast? Twice of this, 200 mm. Cool. Now, where we use this? Which kind of proteins is used in axonal transport? Of course, this is a cool question. And the answer of this cool question is kinesin. Kinesin works with axonal transport. Cool. But what about reverse axonal transport? Oh, for reverse axonal transport, you need dynein. Okay, let's go with the dynein. Cool. We know about this as well now. The dynein is supporting reverse axonal transport. Cool. Let's about the ciliary motility. What is the ciliary motility? This structure, remember? So this structure is ciliary motility. The rotation. Okay. So what happens? What's this whole story? Why we need this ciliary motility? We need this ciliary motility because we want to develop in normal babies. I will tell you what happens. During our embryological development, the rotation of your internal organs happen. The heart is usually developed on the right side, but naturally it goes on the left one. If it does not happen like this, we can call this cetus inverters. We don't want that. We want norm. Cool. So this movement of your organs happens in your embryological age, and this happens with the help of ciliary bodies. Cool. What about lungs? Well, in lungs, the sputum is being cleared, and if the sputum is not cleared, we call it bronchiectasis. Ectasis means stays. Broncho means lung. So something is staying inside the lung. What is it? It's sputum. Cool. What about your sperm? Well, the sperm moves with the help of ciliary body, a filament on the back of it, the tail. Cool. This is sperm. This is fallopian tube. How the egg travels? The egg travels with ciliary bodies. The egg that comes from your uterus goes inside your fallopian tube, and here the egg is planted, not implanted, prosta, just staying, and this egg is actually moving from fallopian tube to your uterus. And how does it happen? It happens with ciliary bodies. Cool, we understand this. Movement of chromosome, that's very important example too. Now, why we need movement of chromosome? We need movement of chromosome because we need mitosis. What is mitosis? It means one becomes two. It's how your body develops, your body becomes. It's the process of you from a small being to a bigger being. Okay, from a child to a man or woman, whatever it is. Cool. So these drugs, if we inhibit microtubules, so these drugs, vinblastine, vincristine, paclitaxel, these drugs actually they inhibit microtubules, and if we inhibit microtubules, the movement of chromosomes stops. So this can be a big problem, and this will eventually result in cell death. So we don't want that. And this drug called colchicine, this is used in gout, and this actually inhibits microtubules. And then this is like a side effect of this drug. So this is it. Okay, so this was your microtubules. Cool. Let's study microfilaments. Smaller in size. Remember the number 2. Just actin and myosin. 2 type. That's it. Actin, myosin. You must remember them. They are used in muscle contraction and muscle relaxation and cell motility. Cool. Small size because they are filaments, and filaments are used in bulbs. Cool. Actin, myosin, muscle contraction, muscle relaxant. The cell motility. The movement can be done only when actin polymerization happens. Cool. Actin polymerization. I don't want to explain this process because it's a long process, but it happens like this, that just imagine I'm making this diagram, okay, and this is how these 2 proteins are basically, and this is one protein. So basically what happens is that the structure of this one and this one, it comes closer, this thing comes this way, okay, this thing comes this way, and what happens is that the structure becomes much more close. It's like I'm not really able to explain you, but just understand something called actin polymerization happens. Cool. So for the movement, for the cell motility, we need something called as actin polymerization. Now this process can be done by other bacteria as well. What? Something called as listeria. Oh my gosh, what's it gonna do? Well, this listeria does something which is called as tumbling motility. And what is it? It tumbles. And this organism has something called as LLO, and this is a toxin. This is Listeria lysine O, and we can't contain it because it has this toxin. This toxin keeps phagocytes away. Now, with the help of Act A, what is it, Act A? Well, it's a substance, and this Act A helps this Listeria to perform this function, actin polymerization, and that's how the Listeria is able to walk and even run inside your cells. And because of LLO toxin, we can't capture it, and this is how Listeria stays, So Listeria, with the help of E-cadherins, we will study what is E-cadherins, performs molecule hijacking and zona adherence. Okay, cool. You must be thinking, what is this zona adherence? Okay, cool. Wait. Intermediate filaments, the last one. These are intermediate. So we had big, we had small, and now we have smaller. Not the smaller, this middle one. Cool. What is it? It is just a filament. It is medium in size, and let's see this table. This is a cool table. I love this table. So these filaments are seen. Where are they seen? For example, keratin. It's a filament. Where? Seen as cytokeratin. Seen as in squamous cell epithelium. Squamous cell carcinoma. So these intermediate filaments are actually these are marker filaments. Okay, they mark. They can be used as identity filaments. For example, if I see keratin, I will think about cytokeratin. And actually, we don't see keratin, we see cytoskeleton. And this presence of cytokeratin means there is squamous cell carcinoma. And if there is desmin presence, then it means it's a sarcoma, or it's a cancer of muscles, rhabdomyosarcoma. Cool. If it's vimentin, that is, we will see fibroblast. And where we see fibroblast in the tumor of mesenchymal origin. Cool. Let's see glial fibrillary acidic protein, GFAP, something with brain, fibrillary glial, brain. Astrocytes, something with astrocytes like star, and where are they present? I think they are present at B, at blood-brain barrier. Cool. They are present at glioma, cancer of brain. Astrocytoma, again, cancer of brain. Important because it can be, it's very, popular. That's why it's important. Lamin. Cool. Where are lamin? Well, actually, it's not lamin, it's laminin, but they are present in the nucleus, and they can be, present at premature aging. Now, this is called progeria, and it's a part of Werner syndrome. Cool. So now we know intermediate filaments, now we know microfilaments, and now we know microtubules. Cool, amazing.